I'm Dr. Ahmed Saadi from the University of Al Amid. Today I'm going to talk about the DNA decoding and the power of PCR. So I think most of you are familiar to this technique. And before I start with the lecture today, I would like to just give you a brief introduction or to the general structure of the training today. So first of all, the audience or the target for this program will be, or for this lecture will be the undergraduate students in the School of Life Sciences, and especially in the Department of the Microbiology. So the time for the lecture today will be 45 minutes. And the suggested training, we are going to use a PowerPoint presentation, also some interactive video from the PowerPoint, as well as some of the online application like Google Class. And by the end of the lecture today, we are going to do some assessment for you, and also we are looking for feedback from you to improve our lecture for the future. And before I move forward, I just want to ask, would it be okay to carry on with you in English, or you have some difficulties to understand? If not, يعني ممكن? Okay, all right. So from now we are going to talk only in English. Only for the very difficult terminology, I'm going to explain them in Arabic. All right. So I'm just going here to introduce you to the general aims of our lecture today. So the aim of the lecture is to provide a comprehensive understanding for the polymerase chain reaction and its impact in the modern biology. Then we have the behavioral objectives or aims, and that will be, by the end of the lecture today, hopefully all of you will be able to describe the 5 components of the PCR, as well as explain the step-by-step amplification cycles, and then you will be able to define and distinguish between the main kind of PCR. And finally, you will be able to evaluate the major application of the PCR in our life. And as I mentioned in the beginning, I think most of you have already used this application. So in order to achieve those goals, we have to go through this pipeline or roadmap. So we are going to start with the general introduction to the PCR, in which we are going to know the most common terminology. And then we have to go through the workflow for the PCR, including the DNA extractions, the ingredients of the PCR. And then we have to read the result once we have our reaction ended. So we need to read the result. We are going to use the Joule electrophoresis. Once we have finished with that, we need to know the major kinds of the PCR, and including the real-time PCR, the RT-PCR, and finally, we are going to know about the terminology for the kind of the PCR, as well as we are going to study the applications for the PCR. Just in a brief, the main application, not all of the application for the PCR. So to begin with, before I'm starting with the lecture, I would like to hear from you. So we have 2 questions here. The first one, do you know what the PCR is? Any idea? Yeah, I just want to hear from you. Doesn't matter if it's in Arabic. Okay, can someone raise his hand or her hand and answer? Okay, no worries. Take it easy. So the other question. Okay, I have another question for you if you are not able to answer that one. So what problem does the PCR solve? Okay, okay. So you can ignore both the questions and we move forward to another question. You see this picture. Any idea about this picture? Please participate. Yes? Can you speak louder, please? It is a library indeed, but sorry, can you? Sort of. It is actually not the right, but it is not not well organized. You can describe the situation just chaotic library, but the general idea of this picture, we have a library here, and within the library we have thousands to millions of books. Imagine the amount of books we have here, and imagine you have a problem, and imagine yourself as an indicator, and you are trying to find a sentence in one book with a library of thousands to millions of books. Would that be easy to find it? A single sentence in that library? It is impossible, exactly. Thank you so much. So we need to find a solution to find this sentence or this line. And the same thing we can apply for the human genome. Imagine we are looking for a specific gene or gene fragment within the genome. As example, during the COVID-19, most of you have already got COVID-19, and thankfully, most of you were recovered, okay? So, imagine we are trying to diagnose this virus based on a single strand from the RNA. But we have a genome here. It is impossible to find that single gene. So we need to find a technique. Another example, we are looking, for example, for a needle in haystack. مثلا يعني عندنا أبرة في كومة قش، يعني كوم مهول مال قش وريد نلقى بيه هاي الأبرة الصغيرة. So that would not be easy to find it. So we need a solution. And the solution here, we need to amplify the number of the target gene, or amplify the number of the needle here. Instead of having a single needle, maybe we need 1,000 or 1,000,000 of needles, and then everyone can easily find the needle in the haystack once we have 1,000,000 or 1,000,000,000 of these needles. The same example for the human genome. If we are looking for a target gene or a fragment from the genome, for example, 16S, we can easily find it if we increase the number of the genes or fragment to 1,000 or 1,000,000 of genes. And the solution for that came from Karimulas in 1993. He is rightly awarded a Nobel Prize a decade later for developing this amazing technique. I can describe it as like a golden application for the biology. So he developed that, and briefly I can say this technique, it works as a molecular photocopier. So like a copy machine. Instead of having a single copy within like 5 or 3 hours, we will have 1,000,000 of copies from a single page. This is how it works. So this is the PCR. So the PCR, we are starting with a tiny amount of the DNA, like a single strand, and then we ended with 1,000,000 of strands. That would be easy to study and analyze for the future. Okay, clear? So now we have already seen the PCR technique, but we need to know how it works. This is really necessary. So what we need to get this technique to work? First of all, we need DNA template, because we are starting with a DNA. We can't start with another, for example, another biological material like protein or enzyme. Then we need the machine that will do the reaction, perform the analysis, which called the thermocycler, or here in general, we just call it a PCR. And I think most of you have already seen that or worked with that in the lab, since you are in the 4th stage. And once we have our analysis ended, we need to see the result, because without a technique to see the result, this will be a blinded test. No one can know, is it positive, negative, failure, or success. So the technique we are going to use, called the dual electrophoresis, or for another kind of PCR, like the real-time PCR, we can use, it can like give us the result as soon as the reaction starts. I'm going to explain this in detail in the next slides. So first of all, DNA extraction. So DNA represents the genetic material in our body, and you know maybe better than me about the DNA. So, but that DNA is present somewhere in our body. Can you please tell me where we can find the DNA? Do you have any idea? Can you please speak louder? Yes, okay, blood and tissue. Okay, so, Yeah, that is correct. We can find it in the blood. But where in the blood? Okay, so that means inside the cells, yes? And inside the cells, we have a really important component, organs, called the nucleus. So inside the nucleus, we have the DNA, and we don't have, unfortunately, we don't have only one kind of DNA. We have a variety of DNA. Bear in mind, we have mitochondrial DNA, and that one is located in the mitochondria. Clear? Right? And we also have one advanced DNA called the free DNA or the circulated DNA. Unfortunately, that one is not also inside the nucleus, but that is not our business today. So, as you mentioned, that the DNA is present in the cells inside the tissue. So, what we are going to do first, we are going to make the homogenization in order to break down the tissue and release the cell. This will be the first step, and then we need to do another step, important one, to release the DNA from the nucleus, as we mentioned in the beginning. So the DNA inside the nucleus, and we are going to do the lysis, either mechanical or chemical lysis. After that, we are going to separate our DNA. Thankfully, now we have a DNA. We need to separate it from the rest of the tissue ingredients like the protein, enzymes, and other debris that we are not need them for our analysis today. So we are going only to keep the DNA. We do the separation. On the final step, we will use a filter, and we will going to dilute the DNA. So we are going to keep just our DNA template, which is our target today, in order to do the further steps. So here we have the DNA template. Now, it is not just the DNA template. We are also need another ingredients in order to perform the PCR. One of the most important ingredients, which are primers. And briefly, the primers are a short fragment of the DNA. They are going to anneal to the DNA either in the 5' end or in the 3' end. As you know, the DNA is a double-stranded, so the DNA will break down into a single strand. One strand will attach to the forward primer, and the other one from the reverse primer. This will explain later on. And then we need enzyme. And here we have to look for a specific enzyme, which is the tag polymerase. And it is thermo-resistant enzyme. So that enzyme works in a high temperature. And I think we usually extract that enzyme from a bacteria that resists the high temperature. And this enzyme works in about 100 Celsius. And then once we have the tag polymerase, we need the building blocks. So you know what is the main component of the DNA? You know? It is the nucleotides, exactly, correct. And we need to feed the reaction with the nucleotides. So the DNTPase represents the nucleotides. And once we have these four main ingredients, we need to have a solution to make this ingredient work in a magic reaction. And that one called the buffer that will mimic the real or mimic the situation in our body and allow the DNA to replicate. Okay, so briefly I'm going to explain the main steps in the PCR. So the first step, you can see in the beginning, here we have the DNA. So that one, double-strand DNA. And then the first step of the polymerase chain reaction is denaturation, in which the temperature rises to, you see, to 94 to 98 Celsius. In that step, the DNA is going to break down the two-strand into, the one double helix structure into a single strand. And after that, we are going to decrease the temperature to about 50 to 56. This step called the annealing stage, in which the primers start to work and attach to each strand. As you can see, we have two primers, those little fragments here. they already attached to each strand. Unfortunately, the pointer is not working in the best condition. So once we have the annealing, now we start to the most important step, which is the extension or elongation stage, which is step 3. In this step, the polymerase enzyme will start to work and start building the new DNA. So if you see, we started with one double strand, but we are going to end with 2 double strands. During the PCR, these 3 steps will be repeated to 36 cycles, and by the end, we will have about million copies of the DNA. So we started with a single strand, we ended with million of copies. And here I have a video for you to better understand this technique, but unfortunately the video player is not compatible with the one we have on this computer. We are not able to see it right now, but I'm going to share this lecture with you, and then you can feel free to watch it later on. Okay, this is just to summarize what we're going to see in the video. So here, as I mentioned, we have the double strand. You can see it clearly here. And then we have the PCR ingredients in this square. We put them all in one tube and put them in this machine, little machine here, the thermocycler, and we start the reaction. These 3 main steps: denaturation, annealing, and elongation, and we ending with the new 2 copies of the DNA. Okay, now we have our PCR reaction ended, but we don't know. Do we have the right thing? Is it positive, negative? We can't tell anything right now. So if we end at that stage, we are wasting our time because we have no idea what's going on. We need to see the result. And in order to see the result with the conventional PCR or the standard PCR, we need to use this technique, which is called Joule electrophoresis. And in the Joule electrophoresis, we have 2 main parts. I'm going to say this in easy way. So we have a power supply on the right side, and we have the Joule tank on the left side. So the main component of the Joule electrophoresis is the tray. In the tray, we have the Joule, and in order to prepare the Joule, we do that in the lab based on our experiment. So we use Joule agarose and the concentration that depend on the molecular size of our target DNA. So according to our experiment, so we are going to prepare that. And we also need a loading die. A loading die is a specific die that allow us to visualize the DNA during the migration. And then the DNA ladder, which is a DNA with a known fragment size. We are going to see that later on. And also need a very important component, which is the pigment. And in the past, we have used a very well-known pigment called ethidium bromide. But unfortunately, this has been seized because it is a carcinogenic pigment. But I think it is still being used. I have already used that several times during my study in the UK. But now it has been replaced with the safer pigments that allow us to visualize the DNA, because this pigment goes to the groove of DNA and it is start to fluorescence under the UV light. So briefly, so here the first step of the Joule electrophoresis, we usually take 2 microliter from our reaction, we put them in the Joule, and in the Joule we have wells, and we insert the 2 microliter from our DNA in the wells. And then we provide the power. And one thing important I would like to mention about the power supply. But before mentioning that, I would like to ask a very simple question. Do you know what is the charge of the DNA? Okay, thank you so much. Well done. So it is a negatively charged. And once we apply electrical current, what will happen? The DNA will migrate through the Joule toward the positive electrode. And during that, the agarose gel will work as a sieve. sieve يعني كغربال يعني يستخدم. What will happen? The large fragment will move in a very slow way, but the small fragment will move faster. This is how it works. And then once we have that, so we usually run it for about like half hour to 1 hour based on our experiment. We take it to another machine and it's called the UV illuminator and we can see the result. We have the result like that. Okay, so sorry, I have. Now we have a result and I would like to explain to you how you are going to read the result. So you see here, this is our DNA ladder. You see the one with the really lovely fragment on the left? So this is the DNA ladder, which is a DNA with a known fragment will help us to indicate our fragment because in the first instance, we don't know what is the molecular size to our fragment. So it is not possible to know what is the target DNA without having the DNA ladder. For example, we have this 1000 kp ladder, which is going to separate according to the molecular weight each 100 kp or base pair. So we can easily indicate, okay, for example, if you look for sample A, which is aligning with 700, we can see, okay, sample A is 700 base pair, it is our target gene. And once we have a clear band like this one, we indicate, okay, we've got a positive result. Then we have here two bands which raise suspension. Maybe we have a contamination, non-specific binding for the primers. So this is not the best result in sample B. And then and finally in the sample C, once we have this really faint band here, we can say, okay, our PCR unfortunately didn't work. And we usually run positive control and negative control. This is important because the positive control, it is a DNA that we already knew, it works and will make our life easier to visualize and make sure about the reaction. Because if the positive sample didn't work, that indicates we have made a mistake. That's called a personal error, and we have to repeat it from the beginning. And the negative control is really important because if we see a clear band like that in our negative control, that indicates that we have made a mess here because the negative control should be negative control, and under any circumstances, we must see nothing there, okay? Because if we see something there, what does it mean? Do you have any idea? Can anyone tell me what does it mean if we see a band in the negative control? That indicates contaminations, okay? A contamination somewhere in our reaction. And in that case, we have to ignore all the results and start from the scratch. I don't want to go deeper with that one, but I just would like to mention it. And now I have a question for you. So we have two results here. One, it is a perfect result, and one is a bit messy. Can you tell me what is going on here on the one on the right side? So which one is the best? The one on the left side. Okay. Can you tell me why? Okay. I'm going to explain because I have not mentioned this situation. Sometimes in the degraded DNA, we are going to see a DNA smearing in which the DNA is not going to separate into a clear, lovely bond as on the right side. We will have this DNA smear, and here again, it doesn't considered as a successful result, and we have also to repeat that, unfortunately. We are usually looking for that kind of PCR, but that only in our imaginary world, so it will be difficult to have the same result in the real life. But yeah, finger crossed, it is not that difficult, so anyone can master this technique within a few days in the laboratory, especially for you as an undergraduate student in the 4th year of your study. So briefly here, I'm going to talk about some of the advantages and limitation or disadvantages of this technique. So first of all, it is simple, as I mentioned. Anyone can do it after a short training, then cost effective. It is also cheap. Anyway, yeah. we can find, I think, PCR in any lab in microbiology or molecular biology specifically, because it is cheap, not that expensive, and also it can be done like within 5 hours. So, but we have some limitation. The main one, we cannot see the result only if we have our analysis ended, so we have to wait. And the second thing is the contamination, because we have to do some post-processing, as we see with the gel electrophoresis. So now it is your turn. This is some exercise for you to do. Feel free to scan this code, and you can answer it if you want. You just need to take your phone, scan it, and provide an answer for that. I will consider that as done. I just want to move faster because I don't have enough time today. And then I would like to briefly mention the real-time PCR. The main differences between the real-time PCR and the standard PCR is, with the real-time PCR, we can see the result immediately. So once the analysis is started, we can see the result. We don't need to wait until the end of our analysis, because this machine is connected to computer, and once the amplification starts, we're going to see the CT value from the curve on the computer. So the principle of the real-time PCR is similar to the standard PCR? Okay. No, it is a real-time PCR. I think it is present since 2008, so it has been introduced. It is not completely new, but it is a bit expensive, okay? And the main application for that, or the main differences between the real-time PCR and the standard PCR, here we have two pigments, the cypergreen and the tagman. Those are specified pigments. What's going to happen once the amplification starts? Those pigments will start to be fluorescent pigment, and that will be read from a camera on the machine and will send the result to the computer. The main differences between cypergreen and the tagman, the cypergreen is not specific, because what it's going to do, it will bind to any kind of DNA, the target, non-target, so it would be easy also to mix with the contaminated. The tagman, it is a specific because it is a probe that connects only to the target DNA, okay? And then here we have already covered that slide, and then advantages and disadvantages. As I mentioned, the main advantage, we can see the result immediately once the analysis is started, and we can use the PCR also for more advanced application. We have also a question for you here. Can you tell me that about the DNA? So we just did amplification for DNA. Would it be possible to do the amplification for RNA? Yes, that would be possible, but not in the real world, not in our inside body, because we usually, during the central dogma, we start from DNA, RNA, protein. But here it is a bit reverse, because especially for viruses, we need to start from RNA. This is what happened with COVID-19. So we need to amplify the RNA instead of the DNA. And here with this technique, we can amplify the RNA, which called the reverse transcription. And also it is the same application. The only difference, first of all, we apply reverse transcription, in which we turn the RNA into DNA, and then we carry on with the analysis. This is really important because here we can study the gene expression, as well as we can study the viruses and other important applications. So here I would like just to briefly say what are the main kind of PCR. We have the conventional PCR, the first one we have seen today, then qPCR or real-time PCR. The third kind is reverse transcription PCR, and we can combine the real-time PCR with the reverse transcription, and we ended with the QRT PCR. Finally, I just would like to briefly mention the main application for the PCR. So, for example, during the COVID-19, we have used PCR اللي احنا كنا نسميه فحص المسحة اللي هو it give us a really quick indication about the result. And then another application, we can use it in the research, for example, for gene expression or developing cancer biomarkers. And another lovely application, I would like just to explain that for you. So in the forensic بالأدلة الجنائية نستخدم هذا. For example, the DNA profiling. On the left side, we have to indicate the biological father for this little one. And we can see based on the DNA fragment that the biological father is number 3 because we have exact match in the DNA fragment style. And the same thing can be applied for the crime scene. مسرح الجريمة. We have sometimes a variety of samples. And normally in other countries they have a database for the suspected criminals. عندهم قاعدة بيانات للمجرمين أو أصحاب السوابق. So once they have DNA in the crime scene, they will compare it with the one from the suspected person, and they will come with the result that, for example, here from those 3 persons, the suspected man is that one in the middle. And the final 2 applications for the PCR we are going to see today, one in the agriculture, for example, indicating the pathogens, and the final one, which is in archaeology, for example, that can take us to the past to study the ancient DNA. Finally, just to summarize, today we have talked about the PCR, which include 3 main steps, 4 steps, which are the amplification, DNA extraction, and gel electrophoresis, reading the result, and it has a variety of applications as we have seen. So now, in order to improve our lecture for the future sessions, I ask you kindly to provide a feedback for our lecture. You can easily scan this code and provide feedback. Okay, it will be really appreciated to have that. And I also ask you to submit your homework. Unfortunately, you have to do that on Google Form. You have to scan these 2 codes or visiting the links shown on the screen in order to complete your assignment, the homework. And finally, if you want to do further reading, you can visit those references that have been used to design this lecture today. And finally, I would like to thank you so much for your patience and for being here today. Thank you so much. And if you have any question, please feel free. Thank you. All right, thank you so much. So.